What's up, Grå Hal? Everything okay? Okay, so it's a great honor and a real privilege to be here today. I am very, very honored because I was actually asked if I wanted to do this in DR-byen, but I so much prefer to be here in Grå Hal. Grå Hal is, from my youth, the best place, together with the other places out here in Christiania. And I'm very honored that the Nobel Prize Committee has pointed towards click chemistry as recipients of the Nobel Prize 2022. And I'm also honored on behalf of all my colleagues, all my friends who have worked very hard on making this a really important new thing in chemistry. So when you get a Nobel Prize, the first thing that happens is that your IQ is increased with 30. And for this reason, you have to make some mantras, and here's my mantras. The first mantra is chemistry is everything, and I really mean that literally. Chemistry is not good or bad chemistry. Chemistry is just everything. It's when you get tired, it's when you fall in love, it's when you get hungry, it's the iPad you're sitting with, it's your telephone, it's the cars that we are driving around in, if we have a car. So if chemistry is everything, it's extremely important for us to actually be concerned with chemistry from a very early age. So I think chemistry is just as important as learning to write or learning to calculate in math. And that is why I have the second mantra, use for chemistry. We need to teach our young people more about this invisible visual world that we call chemistry. And then we need to be able to do research without constraint because there's some artistic aspect of chemistry. Something that has to do with the same thing that my friend Henrik is doing down there with his painting. So, so there's an element of art in the way that we perform science. The new things, the really new things require your brain to think outside the box. And then I think that very importantly, we need to have funding by performance. That means that if we do it very well, there's no reason that we should make some blue sky ideas that are never going to succeed and just follow some buzzwords from the foundations around. We just need to get those people who perform very well in the last 3 years, another 3 years to do research because the likelihood they will do something good is very high. I did not receive the Nobel Prize alone. I share it with my very good friends and colleagues. Yes, there it is. As I said, my good friends, Barry Sharpless and Carolyn Bartozzi. We shared the prize in last year in October. And I was very privileged as a child. I'm very often asked, where did the inspiration come from for all this? And I was very lucky to have grandparents that had a farm on Bornholm. This is their meadows. I visited last year together with Henrik, and this is the meadows. And this is what I looked like at the time. Some change. This is the forest that we walked through, and they have beaches as well. And now when you are in this kind of wonderful surroundings, you start wondering, what is it? So I was playing in the fields one day, and I saw this micro-universe under the carpet of the barley, of weeds and flowers and insects and so on. And I really got, around 8 years of age, this epiphany of why is it so beautiful? How does it fit together so well? So that actually inspired me to later on think that how can I explain all of this? I can explain it with chemistry. So that is why I later on, after a education as an engineer and some studies at Copenhagen University. I went to Carlsberg Laboratory. This is Carlsberg Laboratory, and with me in the package, I had this reading of Limits to Growth and Lord of the Rings, two fantastic books. Limits to Growth actually foretold all the problems that we see today with the environment. Limits to Growth also told us about what would the problems be with the limited resources we have. We see all the consequences of that today in terms of wars, in terms of environmental crisis and so on. And we should have known because somebody told us already in the 1980s. So this was one of the things I had in my package. The other one was Lord of the Rings, what is good and what is bad. Very clearly carved out, and I took that with me, and here at Carlsberg Laboratory, I established a center, a research center, which was supposed to do something important in chemistry. And I looked at it, and there's proteins, there's DNA, there's carbohydrates, and so on. And we chose to work with carbohydrates, proteins, and peptides, the most important molecules in the interaction of cells in our life. And we would use something called combinatorial chemistry, which is derived actually from nature, the way that nature develops the immune response, the way that nature has sexual replication. Sexual replication makes you individuals individual because we recombine the genes all the time. And this is what we wanted to do just in a chemistry toolbox. So we worked with quantitative chemical transformations because we wanted to make millions of compounds and test them the same way nature does it. And this was really a game changer. What is it? We take these small plastic beads, they are 0.5 millimeter size, and we couple a reagent to it. So this is a chemistry that we perform on each bead, and you can see we perform 3 kinds of chemistries. We take them out and we mix them together in a big container, and then we divide them out in the small containers again, and we come with a second coupling reagent or reagent that reacts. And now we have made 12 compounds very rapidly. So these are 12 different compounds that we can now test in a bioassay. If we have 3 blocks and 3 steps, we get 27 compounds. And if we have 5 blocks and 5 steps, we get 3,000 compounds, and you can see it very rapidly, exponentially actually grows to millions of compounds. So that's what we wanted to do, and how do we then do that practically? We invented a small container which can do all the mixing, and so it's just like in the bar when you make a black hole. You just shake the container and you put it back in the small columns here, and then you do a split and a reaction. So you start with the coupling over here, coupling, you deprotect, you mix, you split, and you add an enzyme at the end, and then your beads contain something that allows us to look at the activity of this enzyme. So each of these small compounds that we have made here can behave differently with this enzyme, and we can see the result visually because we have something fluorescent attached to it. So we worked with that, and that also was the start of a lot of different things, a lot of different new chemistries that we were looking at the Carlsberg Laboratory. New chemistries that would be able to perform perfectly in this combinatorial method. So we tried to make quantitative chemical transformations with all sorts of known chemical reactions. And then we had our eyes open for anything that could happen, and that was just what happened. So here is a new method that we developed, which was very good for synthesizing complex peptides. We observed that we have this very reactive group here, and then we have this with 3 nitrogens here. N is nitrogen. It looks like a lot of chemistry, but that's because it is a lot of chemistry. So here we have something that is highly active at this point and is protected against reaction here. So we can make these kind of things by reacting one at a time, transforming this and reacting the second, the third, and make very difficult molecules. So we thought, can we use this for something else? And Christian Tornø was a PhD student working in the lab at the time. So here is a alkyne. This is 2 carbon atoms, one here and one here. And they are linked together with 3 chemical bonds. How is that possible? Well, you have one bond that goes like this, and then you have 2 bonds that goes like this and like this. So that's how it works. I'll show you more about this in a while. So this here was what Christian was attempting. He tried that for months, actually, with different kind of groups, here and here. Nothing worked. After 3 months, he came down to my office and said he would rather work at Novo than in my lab. And then I said to him, maybe we should look at what you have done. And what he had done was he was transforming a starting material here into a single product in the presence, which was quite unusual, of this very reactive group. So we thought there's something special about this reaction. We should look more into that in detail. And Christian did that, and then we went to the American Peptide Symposium, where he proudly presented this result. He presented the orthogonality of this reaction with all the previously known chemistry. So now we have a toolbox. This is the old toolbox that was used in organic chemistry, pharmaceutical chemistry, and so on. You can see there's a lot of good tools in here in the box, and we can stitch molecules together, but it's quite messy. We have a lot of solvents. We have a lot, so things that makes a lot of mess in nature. And then we have now also a small, very nice little toolbox here. And it's 23 years ago we did this reaction, and still there's only 3 small reactions in this toolbox, which is an indication of this is very difficult. I'll come much more back to what these reactions are. So this is the QARC reaction that we developed. This is called the SPOCC reaction, and this here is the TCO reaction. These 3 reactions constitute a completely unique tool for biochemistry, for material sciences, for nanosciences, everywhere, and I'll show you how. So this is, for example, 2 drugs that are currently in clinic for fibrosis in the liver or in the lungs. This is dangerous diseases, so we need something to mitigate it. And these compounds can be synthesized on very large scale with quantitative reactions, no byproducts in water. So this is a completely new scenario. Before, it was very difficult to assemble something so complex as this molecule here. You can see there's oxygens, sulfur, nitrogens, more hydroxyl groups, fluorides, and so on. There's a lot of different things in this molecule. Normally, this would be very difficult to synthesize. Here, it's just a few reactions in water. So, what is it that we can do with this click chemistry? We can take functionality that we go out and dig up in a biochemistry or in all our functional molecules we have made in the laboratory over the years. We can just equip them very rapidly, equip them with a small handle, a so-called click handle, and then we can combine them into one molecule. So it's like making a machine that can drive and dig and lever the contents into a truck, for example. It has 3 functions. This molecule that we want to make has 3 functions, and it starts with these 3 guys. We mix them together. We put them into a container with copper 1. They steer around for a while, and then we have made one molecule, and this molecule now have all the properties of the 3 parts. So if we take this molecule, which has a recognizing moiety a cell death promoting moiety here and a transport moiety here, and we have linked those together. So now what we can do is present this to a cancer cell. The cancer cell will be recognized, and the little guy with the knife will simply take this whole thing into the cell. It undergoes what is called apoptosis, or programmed cell death, and it's gone. So that is how we see this application of functions that we can combine with click chemistry. So now we are in Stockholm, and this was a fantastic day. This was the first day in Stockholm. You get to the museum down in the center of Stockholm, and you are then asked to, first you are told what the whole week is about, and then you are delivering a piece of equipment that meant a lot to you during your research. And what you see here, it doesn't look very impressive though, is a small container. It has 20 columns. So it's similar, it's a forerunner to our library generator. And we did libraries in this one. It has 20 columns up here in the top. And I carved it in my neighbor's garage. He had a big lace that I could cut it in. So this is actually something that I made at home, and was quite important to me. So that's now in this Stockholm Museum. And here is a dress. This dress is also at the museum because this is one dress for each field in the Nobel Prize. This here is for chemistry, and you can clearly see on this dress that it was not made by a chemist, because the click chemistry gives a 5-membered ring, and you clearly see here that they are 6-membered rings, all of these. So you also sign on the bottom of a chair, and so all the chairs in this cafe where this goes on are signed with the Nobel laureates' signatures, and I'm very proud to have my signature here. So in order to just get it out of the way, this is what it looks like. This is the Nobel Prize. And this here is the click reaction. So you can see now, and I will ask you to note that we have 3 blue atoms. These blue atoms are nitrogens. Then we have 2 gray atoms. These 2 gray atoms are carbon atoms. Between the 3 nitrogens, we have what is called double bonds, both here, and here we have the 3 bonds that I talked about before. So these 2 are the components that react with each other, and I'll show you how. When you add the copper 1, they form this 5-membered ring, and now we have combined this function with this function. Is that important? No. That's just 2 small molecules that are being combined. Everybody does that all the time, so that's not important, but it must be important anyway because this reaction is used almost everywhere in science. And the reason that is important, it comes in a while. What happens in the click reaction is that you have this triple bond, and this is one of the bags of electrons that you have in the triple bond. So these are the ones that goes like that. And here is 2 electrons in this sac here. So you see 2 electrons in here, and they are of course negative. So they will look for a positively charged counterpart, and that is a copper ion that comes in here. When it sits on these electrons, it sucks out the density, and that makes this guy here mad. It doesn't like it. It becomes acid, or sur på dansk. And when it becomes acidic, it goes off, and the copper takes its place. Doesn't look that much different though, so what is the reactivity all about? Well, this is still negative, and it sees another copper atom that comes in and sits here on the surface. And now something happens, because the second atom, it opens it like a can of sardines like that, so you can get all into the positive charge that is there in the center. So that's what happens. It opens it up like that, and these balloons that you see there, they are empty. We call it in chemistry LUMO. Don't think about that. They're just empty balloons. And they're looking for electrons because we don't like empty balloons. And these electrons, they come here. These nitrogens are very electron-rich. And fortunately, they have balloons that correspond exactly to the ones where they have to make a bond. So now they will do that very nicely. Boom. First to make what we call a metallocycle. That loses one copper atom. Then a second copper atom is lost, and now we have the product that I showed you before. But as I said, that's not very exciting. What is very exciting is that we can use this all over the place. For example, here we have two functional molecules. This here is a molecule. This here is a molecule that can recognize a cancer cell, and this is the one that can transport the molecule across the cell membrane into the cancer cell. And when we add the copper here, we get the triazole, so now they are linked into one functional, multifunctional molecule. Hi?
Can I ask something?
Yes?
Very interesting. This molecule from the cancer is become for the garbage?
Yes. Okay, but we have one issue with this reaction, and this issue is called the redox issue. And that's the only issue that is with this reaction. And it's quite a serious one if you don't know it. When we do the reaction with the copper 1, copper plus, we have a propensity of this copper 1 to become copper 2 and shrink. And that's completely inactive in this reaction. So we need copper 1 for this reaction. And this happened in the presence of oxygen. So what? We can just reduce this back to the copper 1. So add an electron to this one, and then we have copper 1. But no, that's not the answer. And the reason it's not the answer is because we get what is called reactive oxygen species. That's where you eat this Q10 and all of that to get rid of the radicals that is formed here. So these kind of radicals are quite damaging to biomolecules. This is why you try to avoid free radicals in your food, for example. But how do we then avoid that? Well, we avoid the oxygen. We remove all oxygen from our reactions, and we protect our copper 1 here so that it will stay in the copper 1 state. Well, now we know what to do. We then set out to do something important with these reactions. And the first things we look at is obesity or disease-like, when you're too fat and it's almost like a disease. Then you could regulate your hunger, your food intake, and you can regulate that via a receptor called the melanocortin receptor 4. Unfortunately, there are five different melanocortin receptors, and they have different functions. And we just want to hit one of these receptors in order to regulate our appetite. So what can we do? We take a natural peptide that is for this receptor, a natural molecule for this receptor, and we take the important part and make it part of a very rigid structure to see if we can focus the activity towards just one receptor. And that's what you see here. We have made this cyclic compound here, and this is the rigid part of the molecule that is responsible for the activity. Now, this receptor is not, as I said, only involved in obesity, but also in growth and puberty, sexual dysfunction, memory, pigmentation, and your mood. So better if the mood is involved that you don't get, angry when you take the drug. So we would like to have a drug that's very selective for the melanocortin receptor 4. If we can get that, it will give a signal to these proteins that sits on the inside of the cell membrane, and that will then send a signal to the core. And if we look at the cycle here, we can make changes all around the molecule, make longer chains or shorter chains, make changes of the different amino acids. We made 17 of these molecules, and this was very easy because we have the click reaction. And then this is just a very complicated slide. But that's because it's biology. Chemistry is much more simple. So here is our receptor. Our receptor is sending a signal via several molecules into the nucleus of the cell. And then we get expression of a lot of different proteins that are involved in this cell signaling. If we now change the gene in here and put in a fluorescent protein on this gene, a downstream gene, then we can get fluorescent cells whenever we put something in this receptor on the outside. So we did that. We made a construct. This is called a gene construct. It's just a cyclic piece of DNA. And it has a lot of functions that I won't talk about today because that's not important for what I want to say. But what I can say is that it works. This is without any addition, and you can see these green cells, which are the fluorescent ones, becomes more and more when we add more and more of our ligand. So we found that we had one of our 17 compounds was able to stimulate melanocortin receptor 4 almost without its 300-fold difference here in concentration, almost without affecting the other receptors. So that's very good, and that was the end of this story for our part. So now I'm going back to Stockholm. This is the big day for me, because here I was allowed to talk for 100 million people, which is quite a lot. These are the people that we were talking about, Barry Sharpless, Carolyn Bartozzi, and this is me and Phaedra, who's sitting here on the first row. So this is when you give the lecture. Initially, I was a little bit nervous, but here it's going okay. And then at the end, we were all very relieved, so we gave each other a hug. These guys, they look like tanks suited for war. And I guess they sort of is, because they have been surviving for 400 million years. Not these guys, but the species. And you can see they really look like, they really look like tanks here. They have 4 optical sensors that they can sense everything that goes on around. They have like belts underneath so they can move around. They're called the Japanese horseshoe crab, and we use them because their blood is very special. So we capture these, and the hospital use the content of their blood in order to determine whether you have an infection in your blood. So this is the most efficient way to tell today if you have bacteria in your blood. And what it does is it has 2 macromolecules that comes together around the foreign organism and entraps it. And then it sends in these cannonballs, which are really killer cannonballs. So we were quite interested in looking at these killer molecules. They are called targetplacines. And we were looking at the targetplacine 1 molecule in order to understand how it works. And we were using click chemistry for this, because we were thinking maybe we can replace these 2 SS bridges in the molecule. This is a molecule that looks like small round balls. In reality, it's the same as this one, which is a lot of atoms in space. So we tried to link these together with our triazoles instead. And that worked very well. We could make these molecules. And then when we looked at the structure, we found out that they actually make these cannonballs. because we could see that they touch each other like this. So the most toxic part of this molecule is here buried in the middle, and then it's sort of benign on the surface with positive charges. It swims around, and at a certain point, it hits a cell membrane of a bacteria, and this is negatively charged. Because it's positive itself, it will aggregate on the cell surface like this, and then it will open up and make a pore, and through this pore, all the contents of the cell will come out, and the cell will not be able to survive. So here you see all the water molecules that flow out of the cell, together with proteins or whatever else is in the cell. And then, does that work? And yes, it does. These are 4 different cell types: coli, Staphylococcus epidermis, a serious disease in hospitals, Salmonella typhimurium, which is diarrhea on steroids. This here is a Bacillus subtilis. And does it work? Yes, it works well. The smaller these columns are, the better the molecule is. And you can see this natural one, which is called TP1, is quite active here on coli, epidermis, and typhimurium, but on Bacillus subtilis, there's no activity at all. So this could be because these 2 disulfide bonds can be handled by the microorganism, and they can reduce them. We think that's what's happening because our molecules are working very well with Bacillus subtilis. So we can say click is good for antimicrobial peptides because we can maybe remove some of the resistance in the known antibiotics. Here's another project which I am very proud of. This is a project that was carried out by Christian Kofod in his PhD study. And what he did is he tried to express this as a protease. He tried to express this in coli bacteria. And when we did that, you can see the coli was not very happy about it at all. And the reason for that is that a protease is something that cleaves other proteins. So all the cell's proteins were being processed inside the cell, and the cell dies. So you get a very small amount of the protease expressed, and then it just dies out. So what can you do? We decided to see if we could cut this in 2, so we wouldn't have an active protease, an active enzyme. And once it's cut in 2, we would then express them separately in 2 containers, the 2 parts. And we would express them with this click handle here and here. And then we could just go and ask the coli if that was okay. And the coli say, yes, sir. Because we got a lot of compound here, and we could really work with that. And here it is. It looks very complicated because it is complicated. And here you have the 2 click residues sitting. So when we mix the 2 half, they go together like that. They actually recognize each other very well. So they go together like that. They are clicked together, and now we have a protease. Is that active? We try it with this substrate here, which is a substrate that is known for the wild one. And as you can see, our protease is even a little better than the natural one. Furthermore, in this reaction, we get 100% yield of this protease. So it's a really efficient chemistry. Okay, we are back in Stockholm, and this is the day of the ceremonial dinner in the town hall in Stockholm. It's a nice day. This is first the ceremony with the medal, and then we get the medal, and the king take it back again, because you're not allowed to leave with the medal at this point. So that was a bit disappointing. But then later we have this dinner, and this is me and Phaedra. And what you will observe here is that Phaedra is very efficient because she went to this art school and said, you made a dress that I need to have on this ceremonial dinner. And that happened actually in 2 days. They completely redid it to fit Phaedra. So isn't it beautiful? So very proactive, and unfortunately I have to give her away to Prince Philip. There was no choice there, really. So that's it. This here is an HIV virus. The small yellow spots is HIV that tries to get in or out of a CD4 cell. So that's what happens if you get infection with HIV. Now, these HIV particles, they come in via a mechanism which looks a little bit like this. You have 3 proteins here and 2 proteins here. One is anchored in the virus. Then you get interaction with the CD4 cell, so the immune cell. You get a co-interaction here, it gets closer. Then it spreads out, and you get this hexahelix. So these 2 parts go together and form a hexahelix here. That makes a hole in the membrane. In this fashion here, hole in the membrane, boom, and all the DNA and so on of the virus can enter into the cell. So a lot of viruses has this mechanism, so it's quite exciting if we can prohibit or can make it inhibit the interaction here of these helices. So what we would like to do is take the inner part here and then make a protein out of that and see if we can make something that binds even stronger than the natural helices. So that's what we do. We made a design of a template here with a fluorophore, so this fluorescent, and then you have a small molecular scaffold, and then you have these triazoles which link up these 3 helices like this. Very nice. Now we have a protein, a microprotein that we can use in our assays extremely selectively in a chemistry fashion. You can see it's not these helices that we really have. It's a lot of atoms that sits like that. And then you have the 3 green helices, and they sit there on the surface. But maybe we can make a molecule that will replace one of these helices like that. So we make a computer design of a molecule that binds like into all the grooves, sits very nicely on the surface. And then we can use this microprotein that we have made here. We can use that for looking at the binding. So we put that into nanomolar concentration in solution together with our molecule that we have synthesized here on this small plastic bead. It's only 0.5 millimeter across here. And then we come in with the molecules that we have synthesized, the big molecules, and they go around, they find this bead and bind to it, and now it's orange. So that's how that works. Then does it also work in reality? And yes, it does, because here we have the bead, and this is with our molecule. You can see very, very nice fluorescence. It binds very strongly, even at nanomolar concentration of our microprotein. Here's a bead without this thing, and you can see there's no light at all, so it doesn't bind. And here is a natural triple helix, so that's a very good indication that what we have made is much better than what nature made. So now we're back in Stockholm, and I'm very happy that I had this opportunity as well. This is actually the day before what is on the scheme, so it's on the Sunday. But on this Sunday, I got the medal back. Very nice. That was the first thing that was nice on that day. And I gave a lecture for kids. I love to give lectures for kids, so that was the second thing. And the third thing that was very nice was that I got revenge, because this is Victoria, Crown Princess of Sweden. So it's not only click chemistry that is out there, not only our click chemistry, which is called QARC, that is out there. This click chemistry here is by Carolyn Bartozzi. I just want to mention it because it's used for labeling cancer cells all over the place. So you can see the tiniest amount of metastasizing cancer cells by using fluorophores and this kind of chemistry. What happens is that instead of our copper 1 that catalyzed the reaction, she found a way to put strain into the molecule. So it's like a spring that you bend like that, and the molecule forms like a banana shape that is just set for this reaction. And that's what happens here. So it just reacts like that, and instead of having this bent bond for the triple bond, you get now a bent bond for the double bond, which loves to have 120 angle binding. So this here is much more stable than the starting material. So now I lost my microphone. I'll see if I can get it back on. Yeah, can you hear me? So this here is another click reaction that we came on. It's not the one that we got Nobel Prize for, but it's just as exciting or more. And the reason for that is that here we have a very reactive atom, this carbon atom here. It's protected, so we have removed the reactivity for a while. But now if we treat this with acid, it will fall apart and generate a double bonded oxygen here. And this will react with this nitrogen, which again form a very reactive center that react with this carbon. So we can in one go from here, simple molecule composed from amino acids, to this very complex heterocycle. Very, very nice reaction because it's quantitative and it follows all the rules that we have developed for click chemistry. And the reason that it does that is because it is an intramolecular reaction. So it's inside the molecule we react. So it's 100% stereoselective. And what does that mean? It simply means that this here is a hydrogen. It looks like it's pointing out towards us. It might just as well have pointed back into the plane. And it does only this, what we ask it to do. And you could see, oh yeah, you can see here that all these reactions are completely quantitative. They only give us one product. This here is a line with a peak, and each peak on this line represents a product. And as you can see, there's only one peak, ergo only one product. So we can synthesize a large variety of different structures using this technique. I have shown a small number of those we have synthesized here. And all of these structures look exactly like the drugs we take when we have disease. So we are sure that within these scaffolds or these molecular structures here, there will be things that are very interesting for looking at drug activity. Yeah. So here's one example of use of this. So we were looking at the cannabinoid receptors. And the cannabinoid receptors are very exciting. Yeah, I'm back. They are very exciting receptors. I thought it was PET that came by here. No, because they actually are involved in controlling chronic pain as well as psychotropic activities. So if we can utilize this ability with chronic pain and remove the ability to produce psychotropic effects, maybe we have something that is really useful beyond the psychotropic effect. And here's an amine, and here's an electrophilic molecule that reacts. It's an anhydride, it's called. It reacts with the amine, and it attaches this part to that part. That's chemistry. And here's more chemistry. We're now reacting this here with the red part out in this end, and we get two blue parts here in this molecule. Okay, now we are taking this double bond here and we are reducing it to a single bond. Then we can eliminate water and make a very reactive center. That center here. That center can react with almost anything that is having electrons available. So here we have electrons in this aromatic system, and it reacts here, forming 3 cycles. Now we just have a single reaction here at the end, and now we are forming this compound. So why would we form that compound? That's because we know what the receptor looks like. And the receptor with this molecule will bind it like that in the cavity where it's supposed to go. And as you can see, it fills out this cavity extremely well. So it fits almost exactly into the cavity where you normally bind THC. So here it is. And what we want here is to have this response down in this end. So that's something we can do with this new type of click chemistry. We haven't gotten further than this, actually. But we'll see what happens. So if we should summarize all of this, we have 4 different click reactions. This one here, the QAG reaction, the SPOCC reaction. This one I haven't talked much about, but that's probably the one that is closest to be used in cancer treatment. And then this, what I would also call a click reaction, an intramolecular one. They are all controlled by this formula. This is the one formula you should learn your kids, because this is the formula that controls everything in this universe, without exception. So delta G, called Gibbs energy, free energy, and delta H, which is the reaction heat. And then there's something called entropy, which is the degree of disorder which we can have. And the more disorder it is, the more positive this formula is. So you can see it's minus, and this is the temperature. So the less order we have, the better it is. And you know that from your room at home. When you have cleaned up your room on a Sunday and you come to next Friday, it's completely messy. And you have to put in a lot of energy to clean it up again. So that's how that works. So I would like to end up with talking a little bit more about the mantras that I have. Chemistry is everything, research without restraints, funding by performance, and use in chemistry. So we should start early with the children to teach them chemistry. And the reason for that is very straightforward. Kids, when they are born, don't have a language. They learn everything visually. They see everything. They get into a chaotic world. It's completely chaotic. They don't know what it is, and they actually store all the images. They are related, and they are compiled, and they make associations between these images. They even learn language in whole sentences as images, not as words with grammar. So I think we should utilize that ability to dive into the invisible world, the 3-dimensional world of the molecules. This is water. And explain them why water is a liquid. It should be a gas. It's not a gas, it's a liquid, which is very good for us, because without water we wouldn't be here. And the reason that it is because there's a lot going on between the molecules. And we can actually show that. So we can animate the whole process of water. We can distill the water, show them what it is to distill the water, and condensate it again. So they should be taught from the beginning, and then they should be allowed to play. Serendipity is very important in science, so let kids play, let grown-ups play, and get funding per performance, so we can get some new click chemistries and solve some of the global problems with new chemistries. We would like to do that as a big project that we are applying for money for at the moment. where we actually make animations, 5 minutes for first graders every week, 10 minutes for second graders every week, and so on. And if we do that, we get this repository where we have things from the internet. We have made our own as well. And then we have this guy here, and we just call, and then we punch in what we want, great and weak, and we get the video to the class. That would be very good if we could be allowed to do that. And then we can translate to other languages and license out, so we would be independent. Yeah, I think it's very important for us, for political reasons, as well as individual reasons, to learn more about chemistry. Because chemistry is what is in control and will be in control in the future in this world. Chemistry is a fundamental science. In contrast to many other sciences, it's a fundamental science, just like physics, and together those two describe everything. So I would like to end up by thanking all the many, many people who have been involved and still is involved in developing this click chemistry. So I just had 4 fantastic master students that has worked excellently with this click chemistry in order to make completely new molecules with enzymatic activities. And then I would also like to thank my wife, Vidra, my son, Gianni, Anna, and Sandra, who has been extremely understanding and supporting and also contributing to my thoughts about chemistry over the years. So with that, I would like to thank you, Denmark, and you here in the Grey Hall.